Welcome guys to our third video on cellular respiration. So in the last two videos, we explored the reactions that we'll need to understand for cellular respiration. And the previous video, we talked about glycolysis. So the end of glycolysis, we ended up with 2 ATP, 2 NADH, which if we remember, that's going to be the important part. That represents the electrons that we've taken from carbon. They will ultimately go to the electron transport chain. Finally, we've produced 2 pyruvate molecules, 3 carbon molecules that we're going to process and enter into the citric acid cycle. So we'll get a better recap on that with some visuals in a moment, but for now, let's reorient ourselves to where we are in the cell. In our previous video, we finished glycolysis, which happened in the cytosol. We got our pyruvate, and now we're going to enter into pyruvate processing, which will happen just as we enter the mitochondrion. So we'll see in the next couple of slides, our pyruvate will enter the mitochondrion, it will be transported in, and we will process it so that it's ready to enter the citric acid cycle. The citric acid cycle taking place in the mitochondrial matrix, so sort of in the central part of the mitochondrion. Okay, so we're going to see that our carbons are going to be further oxidized as we go. Okay, so let's just again see what happened just as we finished glycolysis. So we started with glucose at the beginning of glycolysis. That's a 6-carbon molecule. Over the course of glycolysis, we split this into 2 pyruvate, 2 3-carbon molecules. That happened in glycolysis. I haven't noted it here, but we did form some ATP and NADH as we went along. Okay, so kind of just thinking about the carbons here. These 3-carbon compounds, these pyruvate molecules, we've got 2 of them per glucose. These are ready for pyruvate processing. So let's go ahead and get into that part of the process. So here is another image of pyruvate. This one we've got all the details laid out. If you remember how we talked about the idea of the more bonds with oxygen a carbon has, the more oxidized it is, you can easily see that this is getting towards the end of its usefulness. Okay, we've already got 3 bonds with oxygen here. Okay, so this is our pyruvate molecule. However, we're going to go ahead and transport it into the mitochondrion as is, and once there, an enzyme complex called the Pyruvate Dehydrogenase complex is going to perform a series of reactions kind of all in one swoop that will prepare us for entering the citric acid cycle. So the first thing that we can see happen is we're going to start generating some of that carbon dioxide. So remember, this is going to represent that fully oxidized state. That means we've used all of it. It means we don't need it anymore. That is exactly why we exhale it and get rid of it. So we're done with that. Okay, so again, our idea, let's kind of think about our process. This carboxyl group is going to be what leaves as carbon dioxide. So we've got our carbon dioxide. So we're also going to be looking at this idea of forming NADH. So this is going to be our electrons that we took from carbon. Okay, so 1 per pyruvate, so 2 per glucose. So we're losing a carbon dioxide. We're also creating an NADH. And the last part of this is to attach a coenzyme A molecule. Really, the way to think about kind of a CoA being added to things, this is really just going to help spring load this. And it's got this bond has a lot of energy. It helps to sort of propel it into the citric acid cycle. So really, it's just going to be there for a second, kind of like the spring that's going to launch it. we're then going to have 2 carbons left. We lost 1 to carbon dioxide. We've got 2 remaining, and we harvested electrons. This CoA got added so that we can propel these 2 carbons specifically into the citric acid cycle. This molecule is called acetyl CoA, where the acetyl part refers to the 2 carbons and the CoA clearly referring to the CoA part of this. Okay, so now let's go back. We've talked about that carbon dioxide, and let's remind ourselves what happened so far. So we started with a glucose. We've now got 2 pyruvate before we enter pyruvate processing. This was just glycolysis. So pyruvate processing, we lose 1 carbon each as carbon dioxide. We also form some NADH at this point. Now what we're left with is 2 carbons attached to a CoA, 2 carbons. So you can still account for all 6 of glucose's carbons. 2 have been used up, and we've now got 4 remaining that we can still oxidize, and that's why we're about to get a lot of payoff. So all of that happened during pyruvate processing. Okay, so taking our 3 carbon molecule, lose a carbon dioxide, gain electrons, attach it to coenzyme A, and it's ready to go. Okay, so this is kind of a separate step. This is pyruvate processing or pyruvate oxidation. So once we've formed this acetyl CoA, we can add our carbon dioxide and our NADH to our tallies total. So now if we are including glycolysis, we've got 2 ATP formed during glycolysis, 2 NADH from glycolysis, plus the 2 per glucose that we got here, 1 each per pyruvate, and 2 carbon dioxides, 1 each per pyruvate. So our per glucose total can be updated. We can kind of keep track of that, and then our acetyl CoA is ready to launch into the citric acid cycle. So let's check out the citric acid cycle. This looks like it's going to be a pain in the butt, but it's really not that bad. Okay, so we're going to start. Here's our pyruvate, 3 carbons. 1 leaves as carbon dioxide. We formed NADH. All of this is that pyruvate processing step we just saw. Okay, we're in the mitochondrial matrix now. Here's our acetyl CoA. It's ready to go. And again, that CoA is just going to give us the energy to enter the cycle. So we are going to be dealing with kind of looking at 1 acetyl CoA at a time, but we know that in reality, we made 2 of these per glucose. Okay, so what we're going to be looking at per acetyl CoA, 2 carbons enter, and as we go around the cycle, we'll see 2 carbons leave. Okay, they won't be the exact ones that entered, but we'll see what happens in a moment. So the idea is per glucose, 2 turns of this cycle, 4 carbon dioxides, and we've oxidized the rest of them. Okay, so I said that kind of quickly, but we're going to have some time to become acquainted with this cycle. Okay, so again, it takes 2 turns to completely oxidize 1 glucose. Okay, mostly because we've broken our glucose in half. Okay, and something kind of important to note, just so that you're thinking about it, there are tons of these citric acid cycle enzymes in the mitochondrial matrix, so it isn't as if it's like one physical Ferris wheel of a cycle. These enzymes are all over the place, so in reality, the 2 acetyl CoAs are probably being processed separately at the same time, but it makes it easier just to look at one at a time. Okay, so the citric acid cycle actually has 8 steps. We will look at them in depth. But just like glycolysis, you can think of these reactions as being a redox step, which we'll note whenever we see NADH, or a new player here, FADH2. We'll know where a redox step has happened. We'll see some phosphate transfer, which we'll know when we see ATP happen. And when a carbon is completely oxidized, we will see carbon dioxide leave. Okay, so we're going to be trying to also keep up with the tallies as we go so that we end up oxidizing fully all 6 of those carbon dioxides. Okay, so those carbons from glucose. Okay, so let's kind of look at this. So this is the citric acid cycle. Here's our acetyl CoA processed and ready to go. And this looks like it could be heinous, all right, but it's really not that bad. It turns out this makes a lot of sense when you break these reactions down into their purposes. Okay, so we're considering this to be step 1, and that's going to be the entrance of acetyl CoA. So let's just focus on step 1. We don't need to look at all of this at once, okay? They each will have its own chance. So again, this CoA is going to help spring load this into the citric acid cycle. All right, so we've got 2 carbons, remembering in the back of our brain that this is happening somewhere else for our second acetyl CoA. But for now, 2 carbons enter the cycle, okay? The CoA was just needed to spring load this, so we don't need it anymore. It's kind of like a launching pad. Goodbye. These 2 carbons enter the cycle as these 2 red carbons, okay? So that's fine. We glued them, all that happened is we glued them onto a molecule called oxaloacetate, okay? So in a minute, we'll be able to see this a little bit more clearly. Oxaloacetate has 4 carbons. Acetyl CoA has 2 carbons. 4 plus 2 is 6. 1, 2, 3, 4, 5, 6. All we've done is kind of glue this onto oxaloacetate, okay? So not that bad. We've marked the 2 carbons that came from acetyl CoA most recently. These guys are going to take their full turn around the cycle. They're not going to leave as carbon dioxide just yet. They're just going to take their full turn. Imagine if you waited in line to go on a ride at an amusement park. You wouldn't want to get booted off halfway through your turn. You want to take your whole turn. The people who have taken their turn will be the ones to be booted off. So that's what these blue carbons represent. These are going to be the ones that leave. And check it out. Conveniently, they already have 3 bonds with oxygen, okay, these carboxyl groups, so it won't be any problem to just boot them off. Okay, you can see that they're already almost all the way oxidized. Okay, so these guys that are going to leave will be labeled in blue. Okay, and I've got the enzyme that's going to catalyze this specific reaction hanging out right here, citrate synthase. So you can see that we're going to be making some citrate. Okay, so let's explore some of these other reactions. All right, so just to kind of, before we get into that recap, step 1, again, I think important to just keep track of the carbon. We had an acetyl CoA plus an oxaloacetate, 2 carbon, 4 carbon, boom, 6 carbon citrate. Okay, so that's pretty much what's happened here conceptually. So in step 2, remembering that we can kind of categorize things as rearrangements or phosphate transfers or redox, where we're going to be gaining electrons. You can see we're not making ATP here. We didn't get NADH. We have kind of moved some things around. All right, this is seriously just a rearrangement. Okay, so you can see it's just moved. All right, this hydroxyl group's moved. Whatever, just a rearrangement to get us from point A to B. Citrate, it's going to be an isomer, this is isocitrate, not a big deal. Okay, so you'll have some time to look at this in your pre-lecture assignment. Okay, so I kind of want to look at steps 3 and 4 together because we're going to see some similarities. Okay, so notice what's happening here. These blue carbons, check it out. This blue carbon is going to leave. It's fully oxidized. That is a CO2 molecule, 1 CO2 per glucose. We're also going to harvest those electrons, 1 NADH, 2 per glucose, okay? So our enzyme, isocitric dehydrogenase, okay? Step 3, we saw these 2 things happen. Step 4, similarly, another CO2 molecule leaves, and we get another NADH. So collectively, we're going to have 2 CO2 molecules leave per acetyl CoA, and 2 NADHs produced per acetyl CoA, which will give us our running tally now. Per glucose, we've created 4 CO2s per glucose in these two steps and 4 NADHs per glucose in these two steps, plus the ones we gained previously, okay, this 2 CO2 we lost in pyruvate processing, we've now accounted for all 6 carbon dioxide molecules. We've got 8 NADH, okay, so we're racking it up here now, okay, really getting to business on that redox potential, and we still have the 2 ATP that we got in glycolysis kind of sitting in the bank for us, okay, or at the bottom of the fry bag, however you want to think about it, keeping those running tallies. So again, what I've got here in the text is going to be per acetyl CoA. This tally I'm keeping here is per glucose, keeping a running tally since we started glycolysis. Okay, so you can double check, make sure you understand where all of these came from. All right. So you can see we've oxidized these carbons. So we're really getting towards the end where we're just going to be collecting those last bits of redox potential, and we're then going to be regenerating our oxaloacetate. Okay, we're actually already halfway through this cycle as there are only 8 steps. Okay, step 5 is neat. Okay, so take a look at what happens here. So we're going to again be attaching one of these CoAs. This CoA is, again, this is a very kind of an unstable bond, and so as we attach and then allow this CoA to leave, that gives us the energy to create an ATP. Okay, so this says GTP here. That's because our first product we make is GTP. That's fine, we're going to count it because either the GTP can be used directly or it can be converted into ATP so that we can use it just like an ATP. So I'm going to count it as an ATP, right? So we directly make GTP, can either use it as is or create an ATP from it. So 1 ATP produced per acetyl CoA. That gives us 2 per glucose in this step, plus the 2 we had previously, 4 ATP. We've still got 8 NADH. We're done with those carbons. You won't see this number move again because 6 CO2 is in that balanced reaction. Okay, 6 carbons from glucose, 6 carbons gone. So we've got a little bit more NADH to collect as we move around the cycle. We'll also see some new players, FADH2, and then we'll be completely finished. Okay, so let's check out step 6. So step 6 is where we see this new guy, FADH2, come in. So FAD, just like our NAD+, can transport 2 electrons. If NAD+ was our electron taxi, think of FAD as like an electron Uber. So it's the same difference, just a different molecule. We can still transport 2 electrons. So we're going to go ahead and count it towards our electron haul. There will be a little bit of difference between these 2 molecules when we get to electron transport, but for now they are carrying identical cargo, 2 electrons. Okay, so 1 FADH produced per acetyl CoA, okay, 2 per glucose. Our running tally now, 4 ATP, 8 NADH, 2 FADH2, and again, that carbon dioxide is done, okay? So just a few more steps. All right, we're gonna see one more redox step and 2 rearrangements. So let's get going. Step 7, just a rearrangement, okay? So we're gonna see at this point, we still had labeled those 2 carbons that came in as acetyl CoA, but note that this molecule is super symmetrical. Okay, so you can flip it, carboxyl group, CH carboxyl group. If you flipped it, would you know which end is which? You wouldn't, so we stopped labeling them at that point. Okay, so we don't know which is which, but we do know that they're both, these 2 ends are both going to leave at some point. Okay, so here we're doing a rearrangement, fumarate to malate. Step 8, we get our last batch of NADH produced. So this is a redox step. All right, so we've got NADH. We're going to get 2 of those per glucose. All right, so 4 ATP total, 10 NADH, 2 FADH2, 6 carbon dioxide. So this is our running tally per glucose since we threw that glucose into glycolysis. Okay, and so we're almost done with the citric acid cycle. So here we're going to have, again, a redox step, but also we've regenerated that oxaloacetate. Okay, so that's cool too. All right, getting that last bit, and that is step 8. So we are finished. Our last kind of end of this cycle is just going to be to accept another acetyl CoA. So we finished with these. Notice that one of these carbons was red. We don't know which one it is as we go, but one of them is now blue. Okay, so these are both blue, but one of the red ones is going to be blue. The other one was a black one before. So now the guy that took its turn all the way around is going to be one of the guys that's going to leave this time. We'll get a fresh batch in, and then the guys that already had their turn can leave. Okay, so again, this is happening, probably both acetyl CoAs being processed separately, simultaneously, but we've fully oxidized all of the carbons. We've pulled all the electrons off, all the electrons that we can get currently sitting on NADH and FADH2. Those are our electrons that we're going to put in electron transport, 4 bonus Fry ATP, 6 used up carbons. Okay, so in chapter 10, we're going to be talking about photosynthesis, and we're going to talk about how we can regenerate the energy that carbon can give us, why CO2 isn't the end. For now, let's wrap up our citric acid cycle. So now we should know a little bit more about this, okay? So we've gone through glycolysis. We started with glucose. We got a little bit of ATP through substrate level phosphorylation. We got pyruvate, 2 pyruvate molecules, and we got some NADH. Those are going to go to the electron transport chain. The citric acid cycle, we took that pyruvate, took it through processing in the mitochondrial matrix. We ended up with acetyl CoA. Acetyl CoA entered the citric acid cycle, took its turn around. We finished oxidizing all of those carbons. They left us carbon dioxide. We exhaled them. And then we took the electron payoff that we got. And you can see we got a metric crap ton of payoff here compared to glycolysis. This is really the payoff part. We take those electrons and we drop them in electron transport. So we have seen oxygens around, okay? Like there's oxygens involved in these molecules. But we actually haven't used molecular oxygen yet. So when we talk about oxygen, getting these electrons, that O2, we still need to use some of that O2, that reactant. We're going to see how that comes into play in the electron transport, which we'll talk about next week. So for now, we should feel really comfortable with at least the types of reactions that we're going to see, the important payoff molecules that we get, and where these reactions take place, and what we're going to be prepping for at the end. So see you guys next week with our electron transport lecture.